Hi, good afternoon. I hope that you are all enjoying your time at CAR so far. My name is Ashley, and today I'm going to be talking about the bias in self-collected anal specimens and its impact on prevalence estimates for HPV in gay, bisexual, and other men who have sex with men, also known as GBM. I have no disclosures to declare. So human papillomavirus, or HPV, is the most common STI in the world, with hundreds of subtypes that have currently been identified. There are several vaccine preventable as well as oncogenic subtypes that have been linked to anal, cervical, and other cancers. HPV, while usually asymptomatic, can have long-term impacts in high-risk populations, such as those living with HIV and GBM. In fact, GBM living with HIV can have a 5-times-fold higher incidence of anal cancer compared to GBM living without HIV. So recently, Canada has started creating targeted vaccination programs within its provinces for GBM. And this is a perfect time to monitor HPV prevalence at a national level, as this can help to measure vaccine impact and impact of this vaccination program. However, when we're producing these prevalence estimates, they may vary depending on how specimen validity is defined. So to give you more insight into the testing process, I invite you to look over to this graph on the left. So as you can see, the first thing that happens is the individual self-collects their sample, and it's then sent for lab testing, and we use the Roche linear array. And this sample is tested for two different things. The first is the presence of HPV DNA, and the second is the presence of beta globin, the marker for human cells. So looking through the literature, it does not really seem like there's a consensus on how validity is defined. And we found that there's two main definitions based on these two things that are tested for. If you look over this table on the right, you can see the different options that are available. So the first is the definition 1, which says that any sample with HPV DNA or beta globin is considered valid. So in this case, the samples that have HPV DNA and beta globin, the samples that have just beta globin, and the samples that have just HPV DNA are all considered valid under this definition, which is what we're going to call definition 1. And a subset of this group would be definition 2, which only looks at at beta globin positive specimens. So that includes this first row over here that has HPV DNA and beta globin, and the second row over here, which only has beta globin. We can see kind of highlighted this third row is the group that gets excluded when you're moving between definition 1 and definition 2. So the goal of our study was to understand the impact of the way that we define specimen validity on the HPV prevalence estimates that we are getting. So the data for our project is coming from the ENGAGE study. ENGAGE is a mixed method cohort study conducted in Vancouver, Toronto, and Montreal. We recruited 16 and older cis and transgender men who reported having sex with another man in the past 6 months. And we used respondent-driven sampling, which is a form of snowball sampling. Participants completed a questionnaire and self-collected samples for testing. ENGAGE HPV, which is the sub-study that we're working on, started only in men 30 and under, so that's who's going to be in our sample. Now here we're going to talk about the self-collection of anal specimens. So on the left, you can kind of see a portion of the visual representation of instructions that were given to the participants, along with written instructions and some verbal instructions by the study coordinator. So they were asked to insert a swab 4 to 6 centimeters into the anal canal and rotate it 2 to 3 times, touching the walls of the rectum as they pulled the swab out. There was an evaluation of very similar visual self-collection instructions in GBM, and they found that the instructions were essential and easy to follow. And they also self-reported that their understanding of the collection procedure was very high. So these instructions were validated within this population. For our analysis, we are going to be presenting prevalence estimates for both definitions and 95% confidence intervals of detectable anal HPV infection. The prevalence estimates that we are presenting are going to be pooled by study as well as weighted to account for the sampling methodology. The 4 main prevalence estimates that we are interested in are any HPV detectable types, so that includes 36 different types, any of the nonavalent or quadrivalent vaccine preventable types. So there's 2 main vaccines that are authorized for use in Canada currently. The first one that came out was a quadrivalent vaccine, which prevents for these 4 types over here in this chart. And more recently, the nonavalent vaccine came out and is almost exclusively used now within Canada. And the last outcome that we're interested in is HPV-16, which has been linked to anal cancer. So into our participant characteristics, we can see that about 45% of our participants were between the ages of 27 to 30, whereas 55% were between 16 and 26. A big proportion of our sample came from Montreal at about 42%, followed by Vancouver at 32%, and then Toronto at about 27%. Over 80% of our population self-identified as gay compared to other sexual orientations, and 6% of our population was living with HIV, and over 18% reported being diagnosed with an STI in the last 6 months. So if we look at this bottom right quadrant, we can see that no matter which definition of validity was used, either HPV and beta globin or just beta globin, that our proportion of invalid specimens was quite high. So we can see that 76.2% of our samples included either HPV DNA or beta globin, whereas if we only look at those that are beta globin positive, this number drops to below 50%, with only 47.5% of samples including beta globin. What does this mean for prevalence estimates? So here, just to orient you to this figure, we have the black boxes which represent definition 1. So that includes, again, beta globin and or HPV, whereas blue boxes include just beta globin. And we can see that no matter the definition we use, there is high prevalence of any HPV type within this sample. Specifically, for definition 1, we can see that 2/3 of individuals had any HPV in their sample. When we're looking at nonavalent types, this drops down to just over 1/3 of individuals. And then quadrivalent type was just over 25%. And HPV-16, which again is linked to anal cancer, was just over 10% of our sample had HPV-16 detectable within the specimen. In terms of the differences between definition 1 and definition 2, we can see that definition 1, the black boxes, produce higher estimates overall, which makes sense if we think about it, because the group that is being excluded between these two definitions include people who have HPV DNA, but no beta globin. So every single person that was excluded would be positive for an HPV type, which is why there is quite a big difference between some of those estimates. So what are the key takeaways from this study? Well, regardless of which definition of validity that we use, anal HPV prevalence was high in this population. The weighted prevalence showed between 50 to 67% for any HPV type that was detectable. Specific to our study, due to the non-trivial proportion of beta globin negative specimens, and again, for us, we only had 47.5% of specimens that had beta globin, so majority did not. Point prevalence estimates were markedly higher for definition 1 compared to definition 2. Therefore, we think that our estimates from definition 2 are likely closer to the underlying population prevalence of anal HPV infection. So how does this compare to other literature? Well, unfortunately, not a lot of studies are very specific in reporting their beta globin percentages, so it can be a little bit hard to ascertain. However, there was a similar study conducted on a smaller scale in Vancouver that used SelfCollection, and they found that a proportion of samples of beta globin in their study was 66.5% compared to ours, which was 47.5%. There was another GBM HPV prevalence study using staff collection, not self-collection, and they showed a much higher proportion of beta globin positive samples at 86.7%. So what are we doing about this? Well, we have been able to take some action as we identified the problem early on in data collection. So we've been adding markers to swabs to show the depth of which it should be inserted into the anus, as well as using flock swabs to help get more cells from the inside of the anal canal. And we've been evaluating collection areas to ensure that there's a safe and clean surface for individuals to prop their leg up in order to allow for easier access and self-collection. I'd like to thank my team and Dr. Ann Burchell, my supervisor, as well as all the engaged team and participants for all of the work. None of this would have been possible without them. Thank you so much. And I'm looking forward to taking any questions you have in this session below.